The next part of practicals is microscopy. Because as we are observing organisms, some organisms are placed under a slide and they could be already prepared slides which are preserved, or the candidate can be asked to prepare the slides themselves. So this is a plant, this is a commelina that we harvested from the field there. We are now going to work on it and you'll see how you can use it to obtain marks. This is usually done for microscopy, in which the cells of the lower epidermis are observed and drawn and labeled on the board by candidates. But most candidates always find difficulty because they are either not versed with a microscope or they do not know what to do. So first of all, we have to be able to identify the parts of the microscope, because you can't use an instrument you don't know its parts. So this one is a simple compound light microscope which we are using, which is common in most schools and most candidates have access to it. So this one that I have up here, this is the eyepiece, and on it is written 10x, and then down here, what is here, this is what is called the body tube, and here, this is what is called the rotating nose piece. This one you see it rotating, so it is called the rotating nose piece. And then what I am touching here, these are the objective or the ocular lenses. So this one, which is the shortest and which has the lowest magnification, is what is called the low power objective. It has the magnification of 4x, and then this one, which is next to it, this one which is down here, which is next to it, which is a little longer but shorter than the longest one, is what is called the high power objective. So this one here has a magnification of 10x, that is 10 times, and then this one down here, which is the longest, is what we call the oil immersion lens. On the light microscope, it has the highest magnification. So here is written 40x. It means that if you are observing an organism using this small lens, which has a magnification of 4 times, and this eyepiece, which is 10 times, it means that what you are going to see has been increased or magnified 4 times 10, which is 40 times. So this one is what is called a coarse adjustment knob, where you use it to adjust so that you can focus on the organism that you have mounted. Then this one is a fine adjustment knob. We are going to see how we use these knobs. Then this one is the clip. These are the clips that we use them to place the slide on the stage. This is the stage, and then this one is the aperture. The aperture. We adjust the aperture using this knob here. When we turn it, it increases or reduces the amount of light that passes up to the lens. Then this one is the mirror that traps light. So if I turn it this way and it points to the light, it's going to direct light, and the light will reflect and then pass onto the stage, and I'll be able to see. So this one is the handle where we hold the microscope, and this one is the base. So we hold the microscope like this to prevent it falling, and we can tilt it like that to observe so that we don't stand up and we don't strain. So now, to prepare a slide of the leaf of this commelina, what the candidate is required to do is to cut one leaf. He has to make about 2 or 3 trials. He cuts one leaf, he holds it and let the upper epidermis be facing him or her, and then the lower epidermis be facing down. So we're doing what I'm doing. So when you hold it now, what you do is to break it. So you break it from the upper epidermis like that, and then you try to be tearing it. When you be tearing it, you come up with a very thin layer. A very thin layer which is almost transparent. You can see that I've broken it and I have a very small layer here which is almost transparent. This is what I am going to use to make a slide, and then I will observe it. Have you done it? So when I break it now and I have this, this thin layer, what I do is I carefully cut off this thin layer with my hand. I cut it off, and then I put it on the empty glass slide. So when I put it on the empty glass slide now, I'll be required to add water. That is, I irrigate it in water. So I add a drop of water. So usually, a candidate can be given various solutions on which to irrigate the slide, the specimen. So when I add a drop of water, I then take my cover slip. So I take my cover slip and I cover my preparation. So I cover it by starting from one side, getting to the end. So when I cover it, eventually, it lies on top of it like that. So when the cover slip is lying on top of my preparation, then what I will observe is that the water which I had put there is more than what I require. So what I will then do is that I will take a filter paper. This is a filter paper. This one. So I will take the filter paper, and I will take this filter paper and touch the end of the cover slip. So when I touch now the end of the cover slip, you see that it is soaking up the water. All the water which was under the cover slip comes up. It draws the water up, and eventually, as long as I leave the filter paper there, it will continue to draw up the water until the water is finished. You can see how the filter paper is changing its color because it is absorbing the water from under the cover slip. So it draws up the water, and then as it is drawing, you realize that the cover slip now becomes very firm on the specimen. So the advantage of drawing up the fluid which we use to irrigate our specimen is that it also reduces the bubbles, because when the air bubbles are trapped together with the specimen and you mount it, you will see bubbles and you will be thinking that these are cells, but these are only bubbles that are there. But however, when you irrigate it in pure water, it has another biological significance in that the cells will take up water. And when the cells take up water, they will make the cells to be turgid, and as such, the gut cells will bulge out and the stomata will be open. So you will then be able to see the vacuoles of the cells well, and you'll be able to see the open stomach. So after having prepared the slide as such, you mount it on the stage. You mount it on the stage of the microscope, and then you press it with the clips. You make sure that your preparation is directly under the hole, which is there, where light is coming up. Are you there? So when it is under it, you tilt the camera to face you without turning up, and then you trap light. You are trapping light and looking as I'm looking. You are not bending like this, but you sit and you look. When you see that light is shining onto the hole there, you allow it. And then you start your observation with the low power. So when you turn this rotating nose piece and the low power, it clicks. It makes a sound pop. So it clicks, it means that it is already at the right point. So you click it, it goes there. Then you use the quartz adjustment, you take it down till it ends. So when it ends now, you start viewing it with your eye. You can view it with your one eye, and you take it up gradually. You take it up gradually. As you are taking it up, now I can see a few cells. So when I now see a few cells which are not very clearly resolved, I will then use the fine adjustment. So as I'm using the fine adjustment knob, I will realize that it is gradually resolving my preparation, and there's a time I will see the cell walls very, very distinct. So when I now see it as such, under low power, I will change it now. I will rotate the eyepiece, I will rotate the rotating nose piece, and then I will be able to observe it under a high power. So when this one is there, now I'm observing it under the magnification of 10 times 10, which is 100 times. But when I just turn it, I will not be able to see it clearly. So I have to use the coarse adjustment a little bit and the fine adjustment so that I'll see it clearly. So with the higher power, I'm able to see the vacuole, which is well distinct, and I'll see the cell wall. And if I look very well, I'll see the nucleus somewhere, which is at the periphery. And then I will also see the stomata very, very clearly. I'll see the gut cells, how they have made the stomata to be open, meaning that the cells are turgid. So if I could not observe it very well under this one, I could take it also to this oil immersion lens. So when I take it now, I take it down without looking through the eyepiece, but I look on the side. Because if I'm not looking on the side, there's a possibility that this long oil immersion lens can break my preparation. So when it is touching it just slightly, I will stop there. Then I will look here to see what I've observed, and then I will use the fine adjustment knob to take it up gradually, and eventually I will see the cells. So when I see it now, I will locate a group of cells, about 6 cells as the question is usually asked, and these cells are preferably around a stomata. So I will locate it, I will shift my preparation to where I find a very good visible stomata. Then when I see that stomata, I will then focus it in such a way that the stomata is centralized, and then I will also draw the other cells that are by it. So when I observe now these cells, I will draw the group of 6 cells and label it as I'm going to do it on the board. Have you done it?
Yes, sir.
Okay, so I will go now to the board and I will draw a group of 6 cells, including the stomata, which we have observed, and you see how they are drawn and how they are labeled. Oh, this thing is cooler. Okay, I'm on. Okay, I will start by drawing my gut cells. This is how the gut cells were, and I observed that this inner wall here is thicker than the outer wall. And I also saw the vacuole, which is elongated like that. Vacuole, which is elongated like that. So there were many colored things like this inside, many, but the nucleus was very, very distinct. The nucleus was dark, very dark there like that. And then these ones are certainly the chloroplast. So what I have here are the chloroplast. So I will dot the cytoplasm to make sure that this is the cytoplasm that has been dotted like that. So I put the dots on the cytoplasm, and then there were other cells. There were a group of other cells like that, which were there like that. And you notice that these cells are like, some are like squares, some are like pentagons. Some have 5 sides like that. Now the cells were as such. The sizes are something. The cells were as such. Then they have large vacuoles like that. Large vacuoles. So I got the cytoplasm and then I put the nuclei. Nuclei somewhere. Make sure that all the cytoplasms are dotted. So you see that this is a group of cells, and the most concern biologically is the stomata. So what I'll do now is to label it. This one is an epidermal cell. So this is the large vacuole. Then this one is chloroplast. This one is the thin outer wall. Then this one is vacuole of that cell. Vacuole of that cell. This one is the thick inner wall, or thick inner wall of the that cell is the nucleus. And this is the open stomach. This is the open stomach. CH chloroplast. So you could label the vacuoles and all the other ones on any other cell, it doesn't matter. But the stress here is on the stomata. We want to know the various parts of the stomata. What has actually caused the stomata. So you see that this inner wall here, this thick inner wall is thicker, so it gives a lot of pressure and it pushes the other thin wall, which is to the outside, so it bulges out and the stomata opens. So you now realize that we had irrigated our preparation with water, and water is a hypotonic solution. So these cells had taken in water and they are turgid. But usually candidates are given more than one solution. You can be given 2 or 3 solutions. And one of the solutions may be hypotonic, one may be hypertonic, and one may be an acid that will just destroy the cells and you will not see anything. So if you have a hypertonic solution, you will realize that instead of the vacuoles being pushed to the periphery, instead of the cells being turgid, they will be plasmolized. The cells will plasmolize, and you see that the vacuoles will be pushed towards the center and the cytoplasm will shrink. So you will even see that the stomata will close and you will not be able to observe the large vacuoles that we have had here. So usually candidates are advised that when they are observing their preparations, they should be very careful at the differences they note, because there will always be differences, and after the observation, some questions are always asked concerning the observations that we are making. So another mistake that candidates do make when they are writing and when they are concerned with this kind of experimentation is that they do not carry out the experiment themselves. They will pretend and think that they already know. But I want to tell the candidates that solutions are given from any side. You can have an acid, you can have an alkaline, you can have water, you can have a salt solution. And if you have an acid solution, you will not know. All of them will be clear. They will label solution A, B, and C, or solution A and B. So it is advisable to always carry out the experiment and take your time and observe the experiment so that you will be able to draw or to write only what you have seen. Don't write what you think, because what you think may not be what is asked, and although it will be biological knowledge, it will not answer the question and you will not have the mark. So I will advise candidates to be careful, take their time, and carry out the experiment.